GenVoy-ILM T Cell Kit for mRNA is a lipid nanoparticle or LNP reagent mix optimized for the delivery of messenger RNA into activated human primary T cells using LNPs prepared on the NanoAssemblr Spark instrument with Spark cartridges. This non-viral delivery method can be seamlessly integrated into standard human T cell culture workflows. Working with the NanoAssemblr Spark instrument and cartridges, the GenVoy-ILM T Cell Kit for mRNA enables researchers to establish a clinically relevant, effective yet gentle method for ex vivo gene delivery, using a rapid and easy preparation of ultra-low mRNA-LNP volumes for gene delivery at the discovery scale. Let's take a look at how mRNA lipid nanoparticles are made on the Spark. Simply add the formulation buffer to your mRNA working solution, and the lipid mix into the appropriate wells on the Spark cartridge. Cap your cartridge and place it into the Spark. The Spark will illuminate green. Make sure the Spark is set to the correct setting and then press the button. The Spark screen will indicate when the formulation is complete. Remove the Spark cartridge. And collect your mRNA lipid nanoparticles and dilute them with formulation buffer 2. Now you can add your mRNA lipid nanoparticles to your activated human primary T cells using a standard micropipette. This non-viral delivery method involves minimal manipulation of the T cells. You can now continue with your normal T cell protocol. It's that simple.